It's me again. It's my pleasure today to introduce Dr. Anastasia Yendiki. Dr. Yendiki is an associate professor in radiology at Harvard Medical School and an associate investigator at the Massachusetts General Hospital, the Martinos Center for Biomedical Imaging. She is the lead PI of the Center for Large-Scale Imaging of Neural Circuits, which is a multi-institutional consortium funded by the NIH BRAIN Initiative CONNECTS program, with the aim of imaging the human and non-human primate brain circuitry across scales. Anastasia received her Ph.D. in electrical engineering and systems from the University of Michigan at Ann Arbor, where she worked on inverse problems of tomographic image reconstruction. She then moved to Martinos Center, first as a postdoc and then as a faculty member. There she developed TRACULA, if anyone used it, the diffusion tractography toolbox in the FreeSurfer package. She has served as the MGH site PI in the Connectomes Related to Human Disease and spearheaded the IronTract Challenge, which is an initiative bringing together tractography developers from around the world to compare and optimize the accuracy of their methods using gold standard postmortem data. Her current interests are in obtaining accurate models of white matter fiber bundles from microscopy techniques, such as anatomic tracing and optical imaging, and developing methods that can take advantage of these postmortem models to infer connectional anatomy from in vivo diffusion MRI. So with that said, please join me in welcoming Dr. Yendiki.
Thank you. Thank you, Zaki, for the kind introduction. I should have written a much shorter bio so that you wouldn't have to read so much stuff. I was like, oh, that was too long. Sorry. I'm sure nothing can go wrong from changing your slides, 30 seconds before the talk. So this is going to be a surprise for all of us what's going to come out here. But I would try to add, remove some stuff and add more of the postmortem imaging that you guys are interested in, including some hot off the press stuff that no one else has seen outside our group. So I started working on diffusion analysis with the aim of developing tools for analyzing in vivo DMRI data. So, to serve sort of the average user of FreeSurfer, right? So for data that people acquire in their studies, neuroscience or clinical studies every day in centers around the world. But then I got interested in how to validate diffusion tractography against ground truth information from postmortem imaging. And so then I shifted more into the ex vivo world. And so now it's to the point where all of my projects are with dead brains, as I like to say, brains that are in their final resting state. fMRI people get that joke more. But ideally, one day I would like to come full circle and be able to bring our postmortem data and the information that we can extract from that data about connections in the brain back into the ultimate goal of developing better tools for analyzing in vivo data. But as you'll see, it's like a very big circle. So still quite a bit to do. So to this day, most of what we know about how different parts of the brain are connected to each other comes from two different sources of information. So classical tools that anatomists have used to figure that out. In the case of human brains, this has been done mostly with gross dissection, where a postmortem fixed brain is frozen and thawed and frozen and thawed iteratively, which makes it easier to peel apart physically, take apart the fiber bundles, and reveal how they travel through the brain. And this has the drawback that it's relatively low precision. You cannot possibly follow and mechanically remove individual axons all the way from their origin to their destination. So it's more for looking at the big highways. And it has low throughput. So it is very time consuming and requires very specialized expertise to be able to do even just one brain. And as you try to image one of these pathways, you might have to destroy other pathways that are crossing it. So it's hard to get a full brain connectome even for one brain with this approach. But for a very long time, this was the only way that it was possible in the human brain to figure out how things are connected to each other. And then the other classical source of information about brain connections comes from tracer injection studies. The plus of these studies is that they're very high precision. So here a tracer is injected in the gray matter and taken up by the axons. And so it's possible under the microscope, after sectioning and staining the brain, to visualize the tracer, to be able to follow the axons from the point of injection to all their different places where they project, all the destinations of their projections in the brain. However, this is also low throughput. So these studies are also very time consuming, very costly as well. And it's also not possible to get a full brain connectome from this. So classically, this could only be done with like a handful of different tracers. So you can only have that information for a few different injection sites in one brain. And then you have to do multiple of these studies in multiple animals to be able to combine the data to be able to figure out a more dense connectome. And this is not feasible in human subjects because this is an injection that has to be done in vivo and then the animal is sacrificed. But to this day, when a neurosurgeon, has to stick a sharp object into your brain and has to figure out, what should I cut, what should I not cut? Most of what they know and the knowledge that guides them in those decisions actually comes from these kinds of methods and these kinds of data. So obviously there was a lot of excitement a couple of decades ago when this method of diffusion MRI tractography was introduced. It has very high throughput. So with just a short, like 10 to 15 minute scan, you can get a whole brain connectome for a single subject. And it is feasible not only in the human brain, but also feasible in vivo. So you could do it in live patients and do it repeatedly to follow them as they progress, through disease or through brain development, aging, etc. However, the big question mark is in its precision. So how much can you trust the connectome that you get with this method? And this is a question that has been very prominent in the diffusion tractography community for quite a few years. So from the very early days of tractography and from the very first comparisons of tractography to those two classical methods, so the tracer injection studies in the macaque or the gross dissections in the human subjects, it has been known that tractography does pretty well in finding these large highways of the brain, the big named pathways like the superior longitudinal fasciculus, the cingulum bundle, the corpus callosum, etc. But it has also been known since back then that there's a lot of details that it gets wrong. And so from those early days, there was a big effort to come up with these protocols for manually at first interacting with the tractography data to isolate these large highways of the brain that we could get correctly and remove any of the stuff that are incorrect. And so these were called multi-ROI protocols because they consisted in drawing a small number, handful of regions of interest in subject's scan and isolating from the whole brain tracts the ones that go through these ROIs or don't go through some other ROIs, etc. So it was a set of rules based on just a handful of ROIs that were drawn in the subject's data manually. So it required a lot of manual interaction with the data, and again, a lot of neuroanatomic expertise on the part of the human operator. So there was a lot of interest from those early days to come up with automated ways of doing that. And so the first automated methods were essentially a variant of that multi-ROI approach, where instead of drawing manually the ROIs in every single subject's brain, now the ROIs live in some template space, and you warp that template to your individual subject's data. And again, use those rules based on that handful of ROIs, rules on which ROIs a specific pathway goes through or doesn't go through, to isolate each of these highways. So this solved the problem of not having to do this manually. So it automated the process, but there is still an issue of robustness. So if the streamlines that correspond to this particular bundle that you're interested in were not included in the whole brain tractography data to begin with, there was nothing that these ROIs could do for you to make the tracts magically appear. So we came up with a slightly different approach that is not a multi-ROI approach, and this is now more than 10 years ago, and this is where I start to feel ancient. So this was first published in 2011, and the tool was part of, I think, FreeSurfer 5.1. So we're in the sevens now, so just if anyone is keeping track. So instead of running the tractography first in an agnostic kind of way, and then finding the bundles of interest second by using some ad hoc ROIs, TRACULA actually tries to include the anatomical information about where these bundles go through in the tractography step itself. And so it tries to generate tracts that fit in a way that fits the shape of the tract, both to the subject's diffusion data based on this likelihood term. So it tries to generate tracts that follow the orientations of the diffusion orientations from the subject's own diffusion scan. And also some tries to fit the shape of the streamlines to some prior anatomical information that comes from looking at the subject's anatomical segmentation that, in our case, came from the FreeSurfer segmentation of the T1 scan of the subject, and using some prior information from a set of training subjects where these bundles were labeled manually, about how likely each of these pathways at every point along their trajectory were to go through or next to, in the left, right, anterior, posterior, etc. direction, each of the structures from the anatomical segmentation. So, how likely am I to be, lateral, go lateral to the thalamus, etc. And so so instead of getting the tracks first and then applying some ROIs, this is a different approach. So when we first did this, we had to use some training data where we labeled the tracts manually in a set of training subjects that used just regular diffusion data that were available at that ancient time. So this is an example of what the superior longitudinal fasciculus, how much of it we could reconstruct from one of those subjects that had a b-value of 700 and I think 60 directions or something like that, which was like a high resolution scan at that time. And then since then, of course, there was a lot of development on the acquisition side, where new scanners and new sequences were developed to collect better and better diffusion data. And we had this Connectome scanner and MGH. So then the idea was, okay, can we use manually labeled examples of these tracts from those very high quality data sets that can only be collected in one of those high-end scanners, and train TRACULA to be able to reconstruct the same tracts robustly and automatically in more widely available protocols. So we came up with a new version back in 2021. So this was a fun lockdown project, getting that paper out. That both includes more tracts than the first version that cover more of the white matter, but importantly that use training data that came from doing the manual annotation of the tracts in the data from the Connectome scanner. And you can see how much more of the superior longitudinal fasciculus, including all three of its components, you can get compared to those older training data sets that we had for the first version. And so in this paper that Chiara published in our previously favored journal, NeuroImage, in 2021, I think this is probably it's one of our last papers there. So she used those manual annotations from the high quality data from the Connectome scanner that included 500 plus directions and maximum B value of 10,000 and 4 shells, I think. Trained TRACULA based on those manual annotations of the tracks from that data, and then each time leaving one subject out, reconstructed the tracts automatically using only the first shell. And then compared that automated reconstruction using that first shell only with a B value of 1,064 or even just 32 directions to the manual annotation of the same tracts using the high-end data from the same subject. And the good news is we found that we could achieve pretty high sensitivity. So the sensitivity here is how much of the manually labeled tract from the high-end data can we reconstruct using the automated reconstruction from the lower-end data. And so we could reach a sensitivity of up to about 90%, both with TRACULA and with the conventional multi-ROI approach. However, for TRACULA to reach that sensitivity, it incurred a much lower reconstruction error in terms of the distance between the automatically reconstructed and the manually annotated tract compared to the reconstruction error for the multi-ROI method to achieve the same sensitivity. So the idea there is that because TRACULA uses that information about the relative position of the tracts with respect to the surrounding anatomical structures, it learns how to find these tracts even in lower quality diffusion MRI data. Whereas the multi-ROI method will basically just take the ROIs, map them onto your subject, and then it really depends on the quality of your subject's data, whether it'll find the tracts or not. So we want now to take this one step further. And so we showed that we could use this approach to do sort of a quality transfer from the higher end diffusion data from the Connectome scanner to a more widely feasible, clinically feasible protocol. But can we use actually ex vivo data where we can go to much higher spatial resolutions than we can in vivo to train TRACULA and then reconstruct the same tracts in vivo data? And this is just a toy example where we did this using two postmortem human hemispheres that were scanned at 0.7 millimeter resolution, annotating the same sub-bundle of the superior longitudinal fasciculus, and using the streamlines as training examples to train TRACULA and learn the relative positions of the bundles with respect to the surrounding anatomy, and then reconstruct the same bundle automatically in vivo data. So this was just a toy example. It was preliminary data for a grant application. And so we hope to expand it much further. And why do we want to go to these high resolutions? Because this will allow us to go beyond these big highways of the brain to the smaller bundles. This is an example of from an abstract that Chiara had at ISMRM a couple of years ago, where she was interested in finding small subcortical bundles that are important in the reward system. And the big bundle, the big round bundle here is the fornix, and it's just shown as reference. You can get it easily at 1.5 millimeter isotropic resolution, as you can at this submicron resolution of 0.76. millimeters. But for all these other smaller bundles, you can only get them at the highest resolution. And actually, the higher resolution data had fewer shells and lower B values compared to the 1.5 millimeter resolution data. Unfortunately, this scan, this sub-millimeter resolution scan, was collected by scanning a single subject 9 times for 2 hours each time. So it's nowhere near being, clinically feasible and scalable to large studies. So instead of having to bring, a student for several 2-hour scans to get these kinds of data from multiple subjects, it would be much more feasible to have an ex vivo protocol that we can apply to a large number of subjects. This is where, the motivation and our general idea and vision for how we can use the ex vivo data to then benefit the analysis of in vivo data in the future. So where do the macaque data come in? As I said in the very beginning of my talk, the most precise method that we have to determine the ground truth of connections is actually anatomic tracer injection studies in non-human primates. So when we cannot rely on prior information about what tracts are there, then we are relying essentially only on the diffusion tractography algorithm. So this is when we want to run the tractography in a completely unsupervised way without having any a priori knowledge of what pathways are there. And so then the big question is which methods for tractography are more accurate with respect to this gold standard of the tracer injections. And so to answer this question, we started a collaboration with Suzanne Haber, who's an anatomist who has been doing these tracer injection studies in non-human primates for decades and has an extensive collection of them in her lab. And so for several brains, after doing the tracer injection and perfusing the animal and extracting the brain, she sent us the fixed brain to MGH, where we collected diffusion data on a preclinical scanner over a whole weekend to get really high resolution data in the exact same brain as the tracer injections. Then we send that brain back to her lab, and there they do all the sectioning and staining to visualize the tracers under the microscope. And then we can co-register the data and compare them, voxel by voxel, the tractography to the tracing in the same brain. And so we used those data sets to compare different methods for both acquiring and analyzing the diffusion data in terms of the accuracy of diffusion tractography with respect to the tracer. And we found that one of the most significant factors that impacted on the accuracy of tractography was deterministic versus probabilistic. Other things like the exact diffusion sampling scheme, like whether it's single shell or multi-shell or DSI, or the exact orientation reconstruction method mattered less than whether you use deterministic or probabilistic tractography. And this was in a study that we published again in that journal in 2021. And it mirrors some studies that were published around that time using different kinds of data that only had information from the tracers about the endpoints, the terminations of the connections rather than the full trajectory, where they also found that probabilistic methods overall were much more accurate than deterministic. So then we made some of those data available as part of the IronTract challenge. And this was another fun lockdown project. So this was a challenge that was done in 2 rounds between 2019 and 2021. And so the teams that participated from more than 20 different institutions around the world optimized their diffusion tractography pipelines, starting all the way from pre-processing to the final result. With respect to their accuracy when compared to the tracer injection study for one specific injection site, and then took that optimized pipeline and applied it to another data set with an injection in a completely different part of the brain. So one was in the frontal pole, and another one was in the ventrolateral prefrontal cortex, to see how robust these optimal parameters were when you moved around in the brain. And so what came out of that study is, first of all, we came up with recommended best practices for analyzing the diffusion data from pre-processing all the way to tractography. And then we also identified some common failure modes. So for all of these different pipelines, including the ones that performed really well and were really accurate with respect to the tracer data, what were the parts of the brain where most of these methods failed consistently? And can we identify some, what's going on in these tricky areas? And so interestingly, we found that those common failure modes across all of these pipelines, even the most accurate ones, had to do with parts of the brain where the fiber configurations diverged from the classical crossing model. So parts of the brain where, instead of the fibers crossing, we have fibers that are branching from one bundle into multiple bundles, fanning, or taking sharp turns. Because our community has been so focused in resolving the crossing fiber problem, and this to the point where this has been sort of used as a synonym to solving all the ills of tractography, because we've gotten really good at that. So that's the good news, because if we hadn't gotten good at that, we would see errors all the way across the bundle, right? But those areas where the errors happen consistently across methods were actually in these parts of the brain, parts of the white matter, where we see fiber configurations other than crossing. So this is one example of where, by looking at doing these validation studies in a few postmortem macaque brains, you can come up with possible ideas for improving tractography more broadly. And we have finally moved that whole validation engine to GitHub. And so now it's possible to download the data sets from the IronTract challenge, apply your own processing, and then you can submit your tractography results and be ranked with respect to the teams, and get your own ROC curve with respect to the teams from the original challenge. Now, it would still be good to be able to do this kind of validation study directly in human brains, where we cannot do tracer injections. Nobody will volunteer for that. So although, the way things are going, maybe you don't need to volunteer for things anymore. They just you can just be forced to do. So in our lab, we have been collaborating with the optical imaging group at the Martinos Center, and specifically with Hui Wang, who is developing a method that's called polarization-sensitive OCT, and can get direct measurements of axonal orientations without needing any tracers or any staining. So what we did to compare directly the results from that optical imaging method to orientations estimated from the diffusion data, and this was a study that was led by Robert Jones, who was a data analyst in our group and now is in grad school. So we took a small sample from a postmortem human brain, and then we performed a high-resolution scan using sort of the same protocols that we used for the macaque brains, but now in this small human sample. And we generated data with different acquisition schemes, again, single shell, multi-shell, as well as DSI. And then we also downsampled the data spatially to get different spatial resolutions. So from the original 250 micron scan all the way down to 2 millimeter scan that is more typical for in vivo. And then we compared the diffusion orientations estimated from each of these diffusion sampling schemes at each of these spatial resolutions, and with different methods for diffusion orientation reconstruction. We compared them to the direct measurements of axonal orientation that you can get with this optical imaging approach in this small area that contained both crossing fibers as well as some of those branching fibers. And so the interesting thing that we found was that the spatial resolution was the most important factor in terms of the impacting on the accuracy of the diffusion orientations with respect to the ground truth orientations from the optical imaging. And specifically, the biggest drop in accuracy was when we went from 1 millimeter to 2 millimeter resolution. And this is kind of a hopeful thing because this is sort of a range where we can maybe hope to push the resolution even in vivo. So somewhere between this 2 millimeter and 1 millimeter resolution, things improve as you go to higher resolutions. And that was more important in terms of the accuracy of the orientations than which specific reconstruction method we used and which sampling scheme we used. And then if you zoom into these areas to see where exactly those errors occur, that improve when we go from 2 millimeter to 1 millimeter spatial resolution, Robert found that actually, again, this had to do with areas where the fibers were not a simple crossing. So these branch points seem to be, also reflecting the results from the tracer injection study from the macaques. These areas seem to be the ones that are trickiest and where you can actually improve things by going to a higher spatial resolution. So we're using now these early data sets where we have both the diffusion MRI and the optical imaging in small samples, because at that time, the optical imaging setup that we had, that Hui's group had, could only image small blocks at a time. And Chiara has been working on putting that data set together across multiple blocks in the same hemisphere and comparing that to diffusion MRI. And so one of the first things that we looked at was some of those big highways of the brain that we had the hardest time getting in the in vivo data when compared to the anatomical literature. One thing that surprised us is that we expected that as we went to these higher and higher resolutions, that we would see more longer connections. Because conventionally we're thinking that the longer connections are the ones that are harder to get with tractography. There are more opportunities to get through tricky areas and stop, etc. So we looked at the superior longitudinal fasciculus, and specifically the more medial component that has some controversies around it, whether it goes between the parietal lobe all the way to the frontal lobe, or whether it stops earlier, more in like prefrontal premotor areas. And so we expected that as we went to the higher and higher resolutions, we would see more and more of the longer projections that go all the way from the frontal to the parietal. But actually, we saw fewer of those, and we started seeing more and more smaller bundles. And actually, if you talk to an anatomist, this is not surprising at all. So if you talk to Suzanne, she'll be like, yeah, we already knew this. But in the diffusion tractography field, we tend to think of these highways as sort of monolithic structures that go all the way from one end to the other, right? From frontal to parietal. Or the uncinate fasciculus, for example, goes from orbital to temporal. However, in the anatomical literature, and specifically the anatomical tracer literature, it's well known that we need to think of these more as highways, where the smaller roads sort of merge on and merge off the highway at different points along the highway, and use these as sort of a conduit to get between different sets of cortical areas at different points along the way. And so this is something that Chiara presented at OHBM last year, and it's in preparation for a preprint. And so she saw that as she went from sort of the regular in vivo resolutions to ex vivo diffusion MRI of the whole hemisphere at, sorry about that, is 750 micron, to even higher diffusion MRI resolutions when we scanned each block separately, and then down to the microscopic scale from the optical imaging data. She saw more and more of those shorter range connections. And something that I'm not showing here is that this agrees with the macaque tracer data, where when looking at the homologous regions and the fibers that travel through the SLF to reach different pairs of these regions. So then we wanted to go beyond just being able to do, a few blocks of tissue over a couple of years, which is how long that the data for that project took to acquire, and it's taking even longer to analyze. That happens, it's okay. So then we were lucky because the NIH BRAIN Initiative came out with this call for a program that's called CONNECTS, or Connectivity Across Scales. And this is one of the so-called transformative projects of the BRAIN Initiative. So the BRAIN Initiative started under the Obama administration, and it was envisioned as a way to develop new technologies for probing the brain. And so the transformative projects were sort of seen as the culmination of all of these efforts within the BRAIN Initiative. And Connect specifically was the last one of these to be announced, where the goal is to get wiring diagrams at microscopic resolutions for the human and non-human primate, and going down to the synapse level for the mouse. So these projects were first announced, the first round of these projects were announced in fall of 2023. So we are now sort of halfway through the second year. And they funded 5 comprehensive centers, 2 in the mouse, 1 in the marmoset, and 2 in the macaque and human, as well as some smaller specialized projects. And we were lucky with 8 other sites to be awarded one of these 2 macaque and human centers. And so this is the Center for Large-Scale Imaging of Neural Circuits, or LINC, that I am leading together with Suzanne Haber, my longtime collaborator who does the tracer injection studies, and Elizabeth Hillman, who is a microscopist who does develops light sheet microscopes. So it's one of these comprehensive centers centered on imaging the human and macaque brain, and specifically a large chunk of the brain that contains projections that are relevant to deep brain stimulation. So these are the projections that go from the motor, premotor, and prefrontal cortex down to a small nucleus that's called the subthalamic nucleus, and that is stimulated with deep brain stimulation for both motor as well as psychiatric disorders. And the goal is working with all these fabulous collaborators from all these sites to get both diffusion MRI data, so starting from the sub-millimeter scale with diffusion MRI, and go all the way down to the sub-micron scale with some novel optical and X-ray microscopy techniques that I'm going to show you in a little bit. And some of this looks like science fiction, but we're hoping to make it reality in the next few years if our funding continues. So the system that we're targeting again is motivated from the field of deep brain stimulation. This is from a recent study from Andreas Horn group, by the way. And you see here that within this small nucleus deep in the brain, there are different subzones of this tiny structure that need to be stimulated to see therapeutic effect for different diseases, going from dystonia to Tourette syndrome, Parkinson's disease, and OCD. And so we know that there must be something different because we're stimulating with DBS, they're stimulating white matter, actually. So we know that there must be something different about the connections of these different small areas within this small structure. And with diffusion MRI, as you can see here, you can get sort of the gross topographic organization of the cortical to SDN connections from the motor, premotor, and prefrontal cortex. And so it's pretty clear that which of these you capture will determine whether you'll get a therapeutic effect. But there's a lot that we cannot see with diffusion tractography, especially with the kinds of protocols that are feasible in patients. And so actually, a lot of what we know about how these small subcortical structures are connected to the cortex and to each other comes from those tracer studies that labs like Suzanne's have published. And this is to the point where one of the widely used tracked atlases for deep brain stimulation from this Peterson et al. Paper from Cameron McIntyre's group was built not using human neuroimaging data at all, because tractography is considered not reliable enough for this purpose. But bringing together anatomists like Suzanne shown here with 3D glasses, who actually traced these pathways, drew these pathways in a human template based on their knowledge of how these pathways go in non-human primates based on their prior tracer studies. So this is sort of one of the gold standard tract atlases for deep brain stimulation in the STN right now. It comes from knowledge, not from human data, but from sort of an anatomist brain mapping the macaque data into human space. So clearly we should be able to do better. And so we need technologies for imaging fibers that can get us as close as possible to the precision of that gold standard, the tracer injection studies in macaques, but that can be applied to human tissue. And so this is what the motivation behind the data collection that we're doing in the LINC project is. And so to get these projections, we have two competing demands. We need both large field of view because we want to be able to follow fibers all the way from the cortex to the subcortex. And we also need ultra high resolution, specifically in these areas deep inside the brain where these fibers are from pretty much everywhere in the cortex are all crowding and funneling through these narrow passages like the internal capsule. If we try to satisfy both of those demands for large field of view and ultra high resolution at the same time, things will get intractable in terms of not just analyzing the data, but even just like storing it and accessing it. And so because of that, we are exploring a multimodal, multi-scale imaging approach that combines the relative strengths of 3 different optical and X-ray microscopy techniques that I'll show you in a little bit, as well as mesoscopic diffusion MRI. So the idea is that by collecting as much data as it is possible, we will be able to figure out how can we scale back and what is the minimum amount of data that we can collect in a brain to still give us an accurate reconstruction of these clinically relevant white matter projections, with the idea that then if we can figure out this optimal undersampled protocol, we can then scale up and do this in more brains. And for example, explore things like individual variability. So for the, let's see, okay, how am I doing with time? Okay, I'm almost there. So for the diffusion MRI, we are using a preclinical scanner for imaging macaque brains and the 3 Tesla Connectome 2.0 scanner for imaging the human brains. This is a scanner, the Connectome 2, that features the highest gradient strength, 500 millitesla per meter, that's feasible in a human scanner right now. And it was installed at MGH just a year and a half ago, thanks to a different project led by Susie Huang. So these are the slides that I added last minute to show you some of the protocols that we've landed on. So we've been, over the past year and a half, optimizing these protocols for both the macaque and human brain. And we have collected the first data sets that we hope to release hopefully by the end of this year. So we have 4 macaques scanned at the moment. And we used, and this is work done thanks to my postdoc, Ting Gong. So she set up a high-res protocol more targeted towards tractography, where she really pushed the spatial resolution to 250 micron for whole brain. And this had a whole scan time, total scan time of 60 hours. And then a different protocol where the goal was not so much to push the spatial resolution, but to collect multiple echoes and multiple diffusion times to be able to do different kinds of microstructural modeling. And so in these kinds of modeling, you try to disentangle the signal that comes from different tissue compartments. So for example, cell soma, particularly when you're looking in the gray matter, or neurite, which corresponds to the dendrites and the axons. And so she has collected these scans in 4 different macaque brains, and is in the process of doing the preliminary analysis. And again, we're hoping to get this out by the end of the year. And then for the human brains, where we these were some of the first scans that we were setting up on the Connectome 2.0 scanner. We had no idea what to expect, but we were actually very pleased. So we had much better results than with the old scanner, which took us, years to try to figure out how to get good ex vivo data from. So this was a high-res scan. This is 400 micron for a whole human hemisphere that they were able to collect during Christmas break. So we could get the scanner to ourselves from, Christmas Eve to New Year's Eve. And it even needed some breaks for the RF coil to cool down every few hours. But we were able to push the resolution all the way to 400, which is something that I had written in the grant, but I felt it was kind of like a stretch goal. I wasn't sure we were able to do this. So by now, we have collected these data sets in 1 more hemisphere. So we'll have these 2 hemispheres scanned with this protocol, and also with a couple of different protocols more geared towards microstructural modeling, including a protocol with multiple diffusion times that we found to be very useful for estimating axon diameter indices, and also a protocol with multiple echo times for doing combined diffusion relaxometry. And again, Ting has set up these protocols and has analyzed these data with the toolbox that she's been developing. So this is kind of like a first, preview of these data that we hope to be making publicly available and publishing the protocols by the end of this year. So then just briefly, what will happen to these brains after we scan them? So this is the method that I mentioned earlier that's called polarization sensitive OCT that Hui Wang has been spearheading at the Martinos Center. And this is a kind of optical imaging that does not require any staining and images the tissue in the block face before sectioning. And the good thing about that is that because you image first and you section second, the images are not distorted. Which is what would happen with polarized light imaging, where you have to first section and then image the slices. So the goal is to achieve 600 micron resolution for this large field of view in the human and in the macaque brain. And then after the sectioning, the slices will go to the team of Zhu Haohu and Elizabeth Hillman, who have been developing methods for clearing and immunostaining and light sheet microscopy for their own Brain Initiative project. And we hope to collect data in a subset of these sections that goes down to 0.6 micron resolution now to visualize individual axons. Because obviously we don't want to have to do this for every single section in this entire volume, things will really get out of hand in terms of the storage and even just like accessing and analyzing the data. We hope to do as much as we can in these first brains and figure out, again, what is sort of the smallest data set that we need to be able to combine the high resolution in a subset of the sections with the lower resolution from the other modalities to get accurate reconstructions of these tracts. And last but not least, we are working with the team of Claire Walsh and Peter Lee at University College London. They have developed, this was their fun lockdown project. They developed this method for X-ray microscopy that's called hierarchical phase contrast tomography. At the moment, it can only be done in this one synchrotron in the world, in France, near Grenoble, near the Alps. And it allows imaging of intact human or other organs. So because it doesn't require staining also, it just requires dehydration. And so they can get resolutions of 10 to 20 micron in whole brain, and then zoom into smaller cylinders inside the brain and go all the way down to the submicron scale for true multi-scale imaging within the same intact brain. And because of that, all these data sets come inherently co-registered with each other because they're all acquired in the same session without cutting the brain. So then we'll have our hands full trying to combine and analyze all these data from these small number of brains to begin with. And so we're in the process of developing methods for cross-model registration, axon segmentation in the light sheet images, as well as multi-scale tractography that combines the information from all these modalities. And then a big challenge is how do you make these data share and in a way that is like helpful for people, because you won't be able to just download these data sets onto your laptop and run a quick analysis, right? So this is work that's being done. The whole data infrastructure work is done in collaboration with a team of Satra Ghosh at MIT. And so there, the idea is to use a web-based interface to share the data that will allow the users to download the data if they want, but also to visualize the data remotely through the web browser. And this is where a lot of the sort of data formats and visualization tools that we know and love for neuroimaging don't actually work, because they are designed for the use case where you can load the entire image onto your computer's memory all at once. But this is not going to be possible with these data. And so here is where we're looking to the nanoscale connectomics community and trying to use the NeuroGlancer tool that has been developed by that community to access data sets online that are stored on the cloud. And we'll have to do a lot of work to build some of just the features that we just take for granted every day when we use our favorite neuroimaging visualization tools, like how to annotate different structures or how to interact with and visualize tracts. So a lot of these things just aren't part of the use cases that these nanoscale microscopy tools have been developed, but these tools are good at accessing these terabyte or more data sets a chunk at a time, and figuring out how to load the data at different scales depending on, so if you're zoomed out, you get a lower resolution version of the whole brain, and if you're zoomed in, you get a high resolution version of just that little chunk that you're looking at. And so Yael Balbaster from the LINC project will be presenting this at OHBM this year. And so we're in the process of still, building that very basic infrastructure to be able to handle the data that the microscopy teams will be throwing our way later this summer. And so I will end here because I'm out of time, acknowledging our funding that is hopefully still there from the National Institutes of Health. I do want to put a quick plug in for our new journal, Imaging Neuroscience. Maybe I'm preaching to the choir here, but as you may know, all the editors of NeuroImage quit our previously beloved journal at Elsevier and started this new journal at MIT Press that is a nonprofit publisher. And 2024 was the first full year of the journal. And we had almost 350 papers published, which is really amazing. And it's a testament to the support of the neuroimaging community that I want to thank on behalf of the editorial team. And I will end by thanking the LINC team. So these are all the pictures that we took when the grant was like getting funded and getting started. So everyone was excited. I realized that I need to make a new slide with everyone looking like exhausted from trying to do all this stuff now and, stressed out from the current situation. So maybe next time I'm here, I'll I hope to show you that slide. So I'll stop here. Thank you for your attention and happy to take questions.
Hey, we'll start with questions. We have some time for like a couple questions now.
Well, any ex vivo protocol questions?
Yeah, how do you get down to a TE of 20 millisecond?
Okay.
I think the shortest we got was like 85
So we
on a
use,
Prisma fit.
first of all, we use 3D EPI.
Yes.
And this is segmented. So we use like highly segmented 3D EPI. That's how we reduce the T. But then, of course, this makes the scan longer. So that's why our scan times are so long.
Do you think we can do it with our scanner?
Um,
How low do you think we can go with our scanner?
Okay, so you probably don't need to go all the way up to like 18,000 B value, right? Because this is kind of what we were targeting here for these, especially for the microstructure ones. So I think if you, for example, if you want a protocol of 1 millimeter resolution and you're willing to stay, below 8,000, then, it's hard for me to say like exactly what T you can reach, but I think you can get to a reasonable TE. But again, my experience is all with 3D EPI. We have never gotten, 2D EPI, even with like SMS, with G-Slider, with all the latest parallelization tools to work well ex vivo for us. So we have not been able to get, good contrast and SNR. So again, yeah, all my experiences from the 3D API. So this is like coming from Carla Miller's old sequence that has, come down the generations, and we're still using some version of that.
I have a question on the problem of kissing fibers.
Okay.
What's a sensible angle that you think would make sense to resolve kissing fibers or branching fibers? Because crossing fibers are easy. So you go at 90 or you go at like 60 or 45, but then what about?
So that's a great question. So I actually don't think it's about the data acquisition and about, pushing the angular resolution or whatnot. Because actually what we do is we take these data and we fit a crossing fiber model to them. So essentially we're reducing all of these configurations to a crossing. And this, these methods are great for it when the fibers are actually crossing. But based on how the current algorithms are set up, no matter how accurate the diffusion orientation estimates are going to be, because when we have an orientation distribution function that, let's say, is going here, and I'm coming in this way, I am going to just keep following the same direction. I'm never going to jump to these other directions. And if I have actually a branching, I would have to do that. So because the methods are set up to give me the right answer when the fibers are crossing, I think that it's not about making the data better. So I really think that it's about going to just a different paradigm from the ODF. Yeah.
Do you think it's just the only way to solve that is priors, or do you think there is anything else?
Hoping not, but so one of my ulterior motives for going to these like microscopy techniques like these is to actually quantify how frequently and where in the brain we have these other configurations that are like crossing versus branching versus turning. And then this will give us the training data that we need to potentially train an algorithm to be able to classify based on the diffusion signals between these different kinds of configurations. So, it might this is like the science fiction stuff, but I think that this is where these kinds of data will be helpful, because right now we don't have even if we use the latest and greatest machine learning methods, we don't have the training data, the ground truth to train them, right?
Okay.
Yeah.
So are you using any, or will there be any transsynaptic tracing happening? And how would you inform tractography with that?
So the tracing still comes from those like conventional, traditional tracers, and it's only used as the ground truth to validate these other methods that do not use tracers. Because ultimately our goal is to be able to do this in human brains where we cannot use tracers. Other projects within the CONNECTS program are working, let's say, on viral tracers that have been very successful in the rodent, but still need a lot of work to be translated and to be robust in the non-human primate. But that's sort of a whole other direction. So we're more targeted towards scaling up the methods that can actually be used in the human.
I'm just curious, it's not really the diffusion part. When you did all of this, did you go back to thalamic nucleus and see how much the experts 3D tracing matches what you are seeing?
So right now we haven't done any of this in like this entire volume. So we don't have the microscopic data in this entire volume. We do have data from Suzanne's lab from the tracers, which is kind of like what they use to guide the development of that tract atlas that I showed you. So we did, using the Human Connectome Project data in a paper that we did with Suzanne's lab back in 2018 in Journal of Neuroscience. There was Safadi et al. We tried to replicate some of the topographic organization that she sees with the tracers, tried to replicate them using HCP data in the human. And we found that for some of these topographic organizations, we get them right. And for others, they're harder to get. So, yeah, go ahead.
So like my question is actually the other way. Is there anywhere that Susan is getting it wrong and the new data at the resolution in humans get it? Because I've worked with that model, like I read her papers and I read the paper in 2018. It's like, that's how we think about Parkinson, like how about all the loops.
Yeah.
But is there now something new that we learned from human that can like actually now we know something better from human, because we always relied on the monkey for human.
Yeah. And, that's why I start the talk like that, because we still rely on those data, right? Even to figure out where to put electrodes in human subjects. So no, we don't have anything more precise than that. Obviously, in the last few years, there have been all these like fancy clearing and immunostaining methods that have been developed, but none of these have made it into like tools in the hands of anatomists, right? And part of the motivation for this project is to find like the most promising of these techniques and try to get to the point where we can do that. But yeah, right now the monkey tracing is the gold standard. Yeah.
That's it. We won't have time for more. So thank you, Dr. Yendiki, for the talk, and thank you for coming.